Welcome. Good afternoon for Dr. Vizcaíno. Good evening, and welcome to this webinar, which is already the second webinar on surveillance. These webinars have been taking place since 2021. And it is a true pleasure to have Dr. Vizcaíno here with us today, because he was actually the first person who gave a presentation here in this series of webinars for this initiative with Dr. Vizcaíno. So welcome everyone, and may I remind you that these webinars are being broadcasted via social media, YouTube, Facebook, and the ICA website. My name is Erica Calderon, and I work for the Inter-American Institute for Cooperation on Agriculture, AICA. And this is a webinar that will focus on passive and active surveillance. Today, we will have very interesting presentations, people who have a lot of experience. And this is an initiative that is the result of the GFTATS. GFTATS, it's an interagency group made up of ICA as the coordinating agency, but also CARIVET, CAFSA, WOAH, CAN, FAO, CVP, USDA, OIRSA, COPEC, and all of you here. All of you who are joining us. This is actually the first webinar of this year of 2024, and it is a true pleasure to be kicking off this session with all of you, and we will be you will be joining many webinars this year with the purpose of preparing the countries for the preparation and response to African swine fever and other transboundary diseases. Without further ado, I would just like to move into the agenda for today. Many experts with very interesting presentations. We will hear from Jose Manuel Vizcaíno from Complutense University, who will be talking about surveillance in general for ASF, active and passive surveillance. We will also have Alessandra de la Cerda from the Official Veterinary Service of Brazil, who will talk about active surveillance in Brazil. We will hear from Fred Soltero. Fred is from USDA APHIS in Puerto Rico, and he will also be discussing passive surveillance in Puerto Rico. Without further ado, I would like to give the floor to the speakers, but before, I would like to remind you that you can introduce yourselves on the chat. Please let us know what country you're joining us from and from what institution you are joining us today. Please select the language of your choice, and please make sure that all questions that will be left for the end of the webinar, all questions can be asked via chat. This can be through the conventional chat or else the question and answer function. Without further ado, I would like to give the floor to our first speaker, Jose Manuel Vizcaíno. Welcome, Dr. Vizcaíno. And just a brief bio for Dr. Vizcaíno. He is a doctor in veterinary sciences for the Complutense University in Madrid. He has done his PhD in virology in Cornell University. When returning to Spain, he developed his scientific and research work as the chief of the Department of Animal Virology between 1985 and 1988, and also on animal health between 1988 and 1993. He is also the founding director of CISA INEA BSL3 between 1993 and 2002, and since 2002, he is a chairperson for animal health at the UCM in Madrid, and also the director of African swine fever and the reference laboratory of the WOAH. He has more than 240 scientific publications on international journals with high impact and 150 competitive projects. He is the director of 38 PhD theses, and that's probably a higher number by now. The scientific work carried out by Professor Sanchez Vizcaíno have focused on studying the immunological mechanism as well as new epidemiological strategies to control some of the main viral transboundary diseases of animals. His research has been extremely relevant to help control and eradicate different viral diseases such as African swine fever, classical swine fever, and African equine fever. With this brief description and bio of Dr. Vizcaíno's work, I would like to give him the floor now so that he can start sharing his presentation and start his presentation. Thank you. Thank you very much, Erica. Thank you very much for inviting me and for this kind introduction. I hope we spend a good afternoon together, and I hope you will all be happy with today's presentation. For me, it is truly an honor to be with all of you. It has been a while since we had a joint webinar, so I am very happy that I was invited. So the global situation of ASF and surveillance and early detection, this is what I will be describing today. You are very fortunate that it hasn't come knocking, at least not very closely. But the truth is that this is a very dangerous disease, which when it arrives into the different areas, the early detection is not as early as one would want to. So what I prepared for you and the agenda for today in these 8 points. First of all, what the global situation is of ASF, which is worse than ever today. I have worked with ASF for many, many years. I'm not going to say how many, because if you think I'm old, then that will make me even older. So I will not say how old I am, but I have been working with ASF for a very long time, and the global situation right now has become worse. Really much worse, and the incidence has become much more significant. So the global situation of ASF, getting to know the virus better and the disease, the transmission pathways and the resistance mechanisms of this disease, early detection, how to do an appropriate surveillance, what the best tool is, the early diagnosis or rapid lab diagnosis, another key tool. What are the most common errors that we observe when it comes to this disease? What is the situation of vaccines in the face of this disease? And to answer, of course, any questions or comments. That is my plan, but I can go faster, I can go slower, as you wish, really. It really is up to you. The global situation is really, really bad right now. We have a number of hotspots in all 5 continents. It is actually the first time in my life that I have seen ASF so widespread. If you see the red there, it is very, very prevalent right now, and it has jumped to the Americas. The jump to the Americas was, of course, of extremely great concern for all of us and for you as well, but also for those of us who are on this side of the ocean. I always think of you in the Americas, and when it arrived here, and for now there is no possibility of controlling it, this poses a great threat. So where did it come from? We never really knew exactly how, but the suspicion is that it had some help of humanitarian aid in many of the humanitarian aid systems that were sent there with contaminated materials. As you know, the hotspot and the outbreak is less than 1,000 kilometers from the USA and less than 600 kilometers from South America. So the situation is really not good. And of course, we're very close to these areas. With respect to Europe, as you can see here, the disease has spread significantly. We started with a lot of it in wild boar and to a lesser extent on pigs, but now it is increasing in pigs. Not very significant values yet. You can see that's 90% versus 10% of wild boar versus pigs. But the situation continues to be very concerning. It's been already a few years, and the situation in Europe continues to be like this. In the case of Europe, the disease is distributed, as you can see, in different areas. We have natural areas with 68.1% of the hotspots. In agroforestry areas, we had 30%, and in agro-urban areas, which is quite surprising actually, in agro-urban areas, we also see this. It is important to see how diversified the disease is. And how can we compare ASF with other viruses? Because we know that this virus is very complicated, and it is becoming very difficult to see and very difficult to obtain a safe vaccine. And here you can see a little bit the numbers. You have a table for you to get an idea. I have included some of the typical viruses that affect pigs. Depending on the number of genes, here we have 170 to 190 versus PRRS, which has 10, parvovirus 2, and PCV2 5. This is the size of the genome, 180 here. We still have some areas to know, but the variant is also very large compared to the other swine diseases, and the number of proteins also affect. And we have like a castle as a structure, because it has a solid structure and it preserves the nucleic acid very strongly. So the resistance capacity is really astounding. It's really astounding. And here we have the COVID-19 virus. You can see it here. And when you compare it, there's practically no color here with COVID-19, and it didn't last very long. We created a vaccine very quickly, and we created a vaccine quickly for pets because people were becoming worried when they knew that their pets could become infected and they could become a factor that could transmit the disease to humans. So it was a period where people were returning their pets. Well, the beginning of the pandemic was really a disaster everywhere because of how new everything was and all that we know. But when you compare a virus with 10 genes with this other virus, we can understand why it is so difficult to deal with ASF and why it was so easy to control COVID-19. So the structure is quite complex and not very well known. However, we have moved forward significantly. It is very resistant in the environment because of that structure. Its strains have a high viral load and mortality. The immune system has no time to respond. It has great genetic and antigenic variability. And there isn't always crossed immunity. Just for you to understand, we cannot have really a universal vaccine for this. We have certain vaccines for different variants, but actually the cross immunity and cross protection is not very easy to achieve because there are many variables. And then there is an immune factor, which is the fact that it does not significantly produce neutralizing antibodies. Most viral diseases induce a neutralizing antibody. And one of the things that we monitor the most is antibodies, because that is what explains best the capacity of response that that animal has. And this is how we realize that in this case, there is no neutralization. There are antibodies, but they are not neutralizing antibodies, and there is no significant amount of these. So this is, in a way, the structure of this virus. With respect to early detection and surveillance, as Erica was saying, you are very interested in the types of surveillance that we have and the ways in which we can have an active or passive system, a syndromic or a mixed surveillance system. In the country where we're working, sometimes we have mixed systems where we bring together the strengths of both systems, of active, passive, syndromic, and mixed. So what is the problem that we face with ASF? Regardless of the technical issues, we have an extra problem. I call it the extra, the Christmas bonus, because it's the fact that no one wants to be the first country to declare ASF. And that's why early reporting is affected. This disease, with cattle farmers, etc., with other diseases, they say, let's be the first ones because it's not our fault. It's just the fact that we need to get things rolling. However, ASF has a stigma that, and I've been working with this virus for so many years, and in many places, you see that early detection where they say this is the first outbreak, or it's the first outbreak that is pointed out, it is the first real outbreak sometimes. And people are afraid. People are afraid of maybe how that people will blame them if maybe they did something wrong to bring it in, etc. So many different aspects that we can discuss later. So how can we do surveillance? And what is the best tool? We must remember that surveillance it does not exist in a generic way. Surveillance needs to be adapted to each disease because not all diseases will have the same entry pathway. So each disease will have different entry pathways, different clinical manifestations, which are different. And of course, this means that the risk of each epidemiological scenario needs to be considered. Unfortunately, for these epidemiological scenarios, there is no one-size-fits-all formula because the epidemiological scenarios can be very variable. And what is very important is to have good information regarding the risk factors, which for this disease in particular, or for any others, there is a significant importance of maintaining those risk factors under control. And training on biosafety needs to be ensured to be prepared for this disease and others. So what are the transmission mechanisms of this virus? There are many, you can see. There are direct transmission mechanisms, animal to animal. As you know that we have two susceptible animals, which are wild boar and domestic pigs. They are the main ones affected by this disease on ASF. ASF, a good thing. I don't have many good news, but this is good news, which is the fact that there are ways in which it is not transmitted, so not like foot and mouth disease, etc. So it has others, unfortunately, and it can be direct and indirect. One of the things that this virus has, because of the anatomical structure and the structure of the virus, which as I was saying, is like a fortress or a castle, makes it extremely resistant, not just in slaughterhouses or in meatpack district, but also in other areas as well. And we have also biological vectors, ticks in particular. We have two types of ticks. And what they do is they amplify the virus. Then the virus is maintained for a very long time in food that is contaminated or food that was made with contaminated pig meat.
And well, one disadvantage of the disease is that a disease like this one should not affect the human species. Well, of course, we are in luck because it's not currently affecting the human species, but at the same time, it is a misfortune for the control of ASF because as it does not affect the human species, many livestock raisers or breeders try to send as many animals as possible to the slaughterhouse because they know that then things are going to get more expensive if they wait. So those animals that come out of a farm that is not duly taken care of, because the truth is they should be all of the animals on that farm should be slaughtered straight away. Oftentimes there's one portion of the animals that are sent to the slaughterhouse. So this is one of the things that we can see in in some countries that have been affected by ASF, that is contaminated products. As the human species is not affected, people are more lenient when it comes to the control of those diseases, and those products are flown over to a different place, or they are put inside a vessel or whatever. And well, that is one of the risks that we end up having because, for example, infected material ends up, for example, in sausages, and at the end of the day, they end up in the garbage, and that garbage may be eaten by a wild boar, and once again, the cycle begins. Airports are not a specific place where the disease may be found. Airports have a tighter control when it comes to removing waste, but that does not apply to ports, that does not apply to highways. People, for example, travel from one country to another. They may be eating a sausage as they are crossing the frontier and then throw out the paper, for example, in a rubbish can that is across the border. People are really not aware of the risk that that entails, but the truth is that the wild boars rummage between the garbage, inside the garbage, they pick up the disease that way. Please remember that just in one drop of blood, 200 microliters of blood of an infected animal, we have over 300 million viral copies. So in truth, the mechanisms whereby, for example, the virus may be transmitted, the vehicles, for example, the fact that one single drop of blood should have 300 million copies, you can see how the virus sticks to the red blood cells. So, well, that's important to consider, okay? So the blood is one of the most dangerous factors for ASF transmission. So you may wonder, well, how do we carry blood inside my farm? Is that possible? Well, there are different hunters, for example, people that go out hunting wearing their hunting boots. They do not realize that they are out there in the countryside. And then when they come back home, they remember about their pigs and they want to, stroke them or give them cuddles or whatever, but they are carrying the virus on their boots. So this is one of the main mechanisms for disease transmission. That is to say, the boots, the shoes that have been in the places where there have been affected animals. Same thing for flies. Flies can transmit little drops of blood through their legs. So there are mechanisms of infection that are numerous for ASF. Contaminated foods are a huge risk because, as you know, they travel a lot. You might think they do not, but the truth is they travel a lot. So at this point in time, what is the main concern that we have in the countries that have not been infected so far? I am going to the main topics that we are currently seeing, or the main issues, and the way in which we might help each other control the disease. As I said, the entry into a backyard or to a farm or to wildlife, for example, that's the easiest way of entry, especially when we are talking about backyards. Why backyards and why wildlife? Because that is where there are remains of human food that end up in these species for the backyard. Pigs, same thing for the wild boars, etc. And there's a lot of contaminated foods. I told you already that there are several countries that have been affected, and those infected or affected countries are sending out products elsewhere. Like, for example, there are sausages and other types of sausages that end up traveling through the borders. So the best protection is really knowing about what ASF is about, having good biosafety, early detection. That's another thing we are going to discuss as well. A proper surveillance program and training for vets and for breeders. It's really important that you train your vets and your breeders. People should really be cognizant about how to suspect the presence of the disease. It's not just about detecting it, but suspecting the disease. If you feel suspicious, you can already put the brakes on the disease. Otherwise, it will be too late. It may be too late. Be careful about rubbish dumps, also paper processing plants, picnic areas, etc., touristy areas. With regard to vaccination, let me talk about it. As you know, we have been working in a project involving a vaccine. It's a DIMA, a vaccine. I am going to share more about it in a few minutes. And we have made quite a bit of progress with regard to this vaccine. So this was the first vaccine that was used to combat ASF in Europe back in 1965. So this was used in Portugal and in Spain, thinking about an attenuated vaccine, that is to say, an attenuated strain, which is based on white blood cells. And when it was used on animals, this was a test, an assay, no problems were detected. And well, because of the huge mortality that was being seen both in Portugal and Spain, encouraged our Portuguese colleagues back then. I'm talking about the 1960s, mind you. So this was the last flask that I found when I was a PhD student. This is the doctor that was my PhD director or tutor. And well, so how did this happen? What was Professor Botija, who was my tutor, or Ribeiro, Mr. Ribeiro, another professor living in Portugal, what was the pressure that they experienced? Well, there was a lot of animal mortality, a lot of cattle mortality, or pig mortality, and both the academics and the scientists were playing around the techniques that worked the most at that point in time. Inactivated viruses, in the case of ASF, they really do not work. We know that for African swine fever immunity, it's necessary to have replication. So they decided to use an attenuated strain. They run a number of assays. So they realized they tried them on the pigs, and it seemed to work. But when the vaccine was applied mainstream on the farm, well, these lesions started to appear. This was because of the positive tick bites that the pigs received. There was also joint problems, inflammation of the joints. This ended up causing necrosis. So there were immunocomplexes all over the place, because this is a virus that has high immunogenicity. So, well, the truth is that these vaccines were not capable of neutralizing these problems. So there were a lot of immunocomplexes. So this originated a number of issues. So what happened as a result of this? Chronic forms of the disease were created, as well as carrier animals. Well, this was an attenuated vaccine. Imagine that back in the 60s, the technology was more backward than nowadays, and the virus reverted quite easily towards virulence. So different forms were originated, so it became more acute and more chronic. So what is early detection? What are the lesions? What is the clinical series of symptoms that may lead you to suspect the presence of the disease? Well, this is a disease that has an initial expression, which is very high fever, and a number of skin lesions that I will illustrate shortly. But quite significantly, what we can find is the lesions that the animal presents, which are very similar to CSF, to classical swine fever, and which present more seriously with ASF. So let's take a look at these lesions. It's not that they are pathognomonic, that is to say, they do not define the disease in and of themselves, not that, but what I can tell you is if you do notice these lesions, that's sufficient cause for suspicion, okay? So, well, probably you've seen these lesions. You have a kidney, you have the splenomegaly right here. It's hemorrhagic splenomegaly, the lymph nodes that have been seriously affected. As you can see, well, the lymph nodes are, well, that's quite flabbergasting. You can see how, well, these lymph nodes or have clots inside. It's as if well, you also have, for example, spleen lesions. You can see the lesions in the kidney. And even though these are not so explosive, you may see some that are more not so noticeable. But you can see the splenomegaly that is almost 100%. So there is, all of these cases are ASF cases. So you should not, if you can see these colors, for example, congestion, hemorrhagia, etc., or bleeding, serious bleeding, well, that's cause enough for suspicion. That is quite significant, and it's really, it's better to be suspicious and take a sample to the lab rather than, turning your back on it and sort of pretending nothing is happening. As to the diagnosis, we work quite a bit on diagnosis. Our lab is one of the laboratories that has developed the most techniques on ASF. Serologically, for example, we adapted an ELISA test back in 1979. I was very, very young back then. Well, this is a virus disease, remember? So the ELISA tests would be used for parasite tests, but it was the first time that we adapted an ELISA test for a viral disease, then IPT, IB, etc. So for the diagnosis of ASF, we really have really useful tools that are highly sensitive and highly specific as well. We also have sampling. We can get samples not just from the blood, but also from the serum and from oral fluids. And we can also do it based on tissues, like, for example, well, of course, blood is one of the best samples that we can get. But, for example, tissue-wise, we can take lymph nodes, for example. The lymph nodes are really good for samples. So what antigen techniques do we use? PCR, and also the insulation of the isolation of the viruses. Well, I'm sure that you have these technologies in your lab, in your own labs, and well, these techniques are working quite well with ASF. What you need to understand is when we are talking about different countries, etc., there is not a single ASF agent. There are varieties that exist. And at this point in time, for those that we have used, we have studied, these tests work. PCR works, ELISA works, etc. So at this point, we have not found serious problems when it comes to diagnosis. Now, Where we do find problems is when it comes to protection. That is slightly different, and we will talk about that shortly. So, well, then we should move on to sampling. Sampling is quite easy because we've adapted this a number of years ago, back in 2021. What I'm going to tell you right now is very interesting. This is a mix of alcohols, very cheap to obtain. You use a small sponge and you need to dip the sponge in the alcohol and dab it on the animal's face, wherever he or she usually sleeps, in the drinking troughs, etc. And not a single drop of blood is used, because if you're going to use a syringe and puncture the animals, and if there is blood, that is going to entail a risk, because the blood is the best mechanism for viral resistance. So in most of the sampling efforts, what we try is to avoid blood as much as possible. Right now, we have a technology that allows us to avoid having to use blood. Well, this is a perfect way of detecting ASF virus, and you just need to dab that sponge in the alcohols and then dab it on the animal's face, etc. And that's that, because from a standpoint of infection, the virus is not infectious with this method, but when it comes to detecting it, it can be detected, okay? So we can talk about this in greater detail later if you want to. Let me talk about vaccines and the failures of these vaccines to date. Well, the first vaccines were the inactivated virus vaccines that did not afford any protection. Same thing for subunit vaccines, same thing for DNA vaccines. The immune response was very weak, no protection was afforded, and the ones that really started working at the beginning were attenuated vaccines. But problem, there was a balance issue between efficacy and security or safety. So sometimes protection was given to 92 to 100%, but the instability of that situation made the vaccine to revert back to virulence, and that happened quite frequently on top of that. So, well, based on Spanish and Portuguese research from 1965 to date. For example, we probably know about these vaccines. This was done with the USDA. There has been no progress from that point on. So you know that these vaccines have also presented issues, different kinds of issues, but we can discuss this later when we are done with the vaccines. So what is the reason why ASF is all over the place? Well, and as part of our project, VACDIVA, when we presented the VACDIVA project, we submitted it to the EU. The EU issued a call for a call for proposals for an ASF vaccine. We decided to run for this call, for this contest, and well, we were given we were chosen as part of this call for proposals. And why did we decide to go for this? Well, because as you have seen, ASF is a devastating disease for wild boars and for pigs. There was no vaccine that was available so far in the market. Currently, there is no DIVA ASF vaccines. We work both on domestic pigs and wild boars, so it's both species. There are several epidemiologic scenarios that jeopardize detection. The disease does not express in the same way in different places, so the scenarios vary quite a bit. It's necessary to have an effective, safe, and DIVA vaccine, a DIVA vaccine, because in a disease such as this one that presents with so many complications, you need to be sure about what animals have been vaccinated and differentiate them from those that have not. This was one of the main issues that I had when I was visiting Costa Rica, and I was close to the rest of the different countries when a decision was made to bring the vaccine over and so on. I said to myself, you're going to have a problem. We need to improve the diagnosis, because otherwise problems may arise. In our opinion, the vaccine needs to be adapted to cell cultures. This is to say that it should not just be manufactured in the leukocytes. The virus works well for leukocytes, and the vaccines produce really high protection levels. But obviously, working with leukocytes entails a problem. You need to have healthy animals, so the cell lines are more stable, they are simpler. So for this reason, we have always worked with leukocytes, of course, but always trying to adapt them to cell cultures. And you can see that we have that.
This is the VACDIVA project. We started working on this in 2020, and we just finished, and we will finish in June 2024, so 4 years. We were given funding worth 10 million euros. It was a consortium of 16 countries, 13 EU, and 3 that were not part of the EU, Russia, China, and Kenya. Russia was with us until the invasion on the Ukraine, where the European Commission decided that they could no longer be accepted, and so they exited the consortium. What are the objectives of VACDIVA that we put into place to produce a safe, DIVA, and effective vaccine for domestic pigs and wild boar? The domestic pig is infected via food or wild boar that jump the fences. This is very important as a factor, and it is dangerous when the disease appears in domestic pigs, because the main factor is wildlife that is near there. So we are working on a vaccine for wild boar, because we believe that the wildlife is the main problem. And put together a DIVA diagnostic method and a vaccination strategy. So to produce a vaccine, to create a differential diagnostic method, and then we have different vaccination strategies in different epidemiological scenarios. All of this carried out by the research groups of the project. The first publication was in 2019, when we did the first oral vaccination of wild boar against the swine fever virus. This was the first step forward with an isolate. So our friends in Latvia isolated this in a wild boar, and it showed very interesting characteristics. So that's why we started using that after that, and this is what we've been working on. What happens is the following: if you include the whole virus, even attenuated, the capacity of a virus to do combinations and reversions are very, very high. Whereas if we work with small pieces of that virus, sometimes we work only with a mutant or 2 mutants, it is less likely for them to create these problems. And if the mutant we choose represents a significant genetic part that is tied to the protection, then we gain a very significant advantage, and we may reach a good vaccine. So what we are doing is testing these 2 finalists after many mutants that we chose. We were left with Delta AB. These are technical names, and the name of the gene, because it's undergoing the whole patent process of Delta AB and Delta ABI8. We've done many tests in INEA, in the INEA here in Madrid, in the NARI, ISUM, NFCSO in domestic pigs, and many others, also in Italy, that people who collaborated with us. So I will show you what the current data looks like in the case of wild boar and in the case of domestic pigs. What we did was work based on the RIE, which is called Latvia 17. It is a naturally attenuated virus found in a dead boar. So several mutants were identified for clinical trials. In the case of domestic pigs, it's intramuscular, and in the case of wild boar, it is oral. The protection that we reach is between 90 and 100%, but we are in the phase of improving this. And with the Delta AB I8, the results have been very, very good so far. With Delta AB, we have seen this in the white blood cells and in the cellular line. And in the case of wild boar, we have a protection between 92 and 100%. And this is in the final phase with no side effects whatsoever with respect to the boar itself, yes, and in the pathology that we have observed in the boar. And we are seeing whether there is a problem in the gestation, but we're evaluating that. But the results are proving to be very, very satisfactory, and we're preparing a test in a controlled environment with Delta AB in a large number of animals so that we can actually see whether there is a problem when this number is very, very high. Until now, we had been working with groups of 60, 90, and what we want now is to have more and for longer. So the conditions supposedly should allow us to have more animals than, of course, in a lab or controlled environment, and for them to be in a safe area. And it has become easier to localize that. So with Delta AB, 92 to 100%, no side effects. The DIVA test is working, and it has been adapted, of course, and prepared by a company that has been working with us in a consortium, and the diagnostic test. So these are the facilities that we have at the university. This is a clinical trial that we are currently carrying out. As you know, these animals are always sleeping. Every time you take a picture, they're always sleeping. Very rarely do you find them awake, but that's where they are. We have done studies to see whether if a vaccinated wild boar with that vaccine, if it could move on to the domestic pig, if this were to happen in a farm or on a farm, because with this system that we've designed, and as you can see there, they have a lot of contact. You can see that there are wild boar right and left. They have a lot of contact. They actually give kisses to each other. It seems like they fall in love. I have never seen humans be so affectionate to one another. So that they have good contact for long periods of time, and all of them were PCR negative, and only one had very, very low levels of antibodies. So there is some kind of transmission, not very high, but if not, we wouldn't have this piece of information. Regarding animals that were vaccinated, even with a challenge and that were slaughtered, you can see that there was no problem in the anatomy or pathological that would make us suspect that they had been affected. With respect to the diagnosis method, we have a kit, a diagnostic kit, and here's a virus, of course, simplified, but it has a part A and part B. Of course, there's more, but just to simplify the drawing. And the vaccine has been added a marker and a part A. So what happens? We have the marker, we have B, and we have A. How do we interpret this? In an infected animal, the marker is negative because logically it's only in the vaccine, but it's positive for B and for A in the virus. Vaccinated animals are positive for the marker and for part A that we have included in the vaccine, but not B. And negatives are just negative in every column. So this very simple chart is allowing us to monitor the vaccination process, and it's essential to know how things are going. In a disease such as this one, I think that the vaccine needs to be marked like this. In the project that we have now, this is something that we're preparing with a high number of animals. In Spain, we have already created a study with administration of decoys in the field. So we have tried to find the best option, the best bait. And the idea is to have a large number of animals and for a long period of time to be able to study whether that vaccination and with a possible challenge could cause problems in the medium or long terms. In Africa, we are doing a test also, but with these bush pigs or these wild pigs or feral pigs that are carriers of the disease. So if the vaccine could help avoid these animals who are carriers, this would be very useful. This is another study that we have done on the bait. The bait was empty here, so there was no vaccine there because, as you know, Spain is free, so we could not vaccinate them. But what we wanted to find was which bait was more attractive to them, which one tasted better for them, which one they actually went after. And we finished this experiment, and the results are very, very good. We had photographs there to show what was happening. We had the entire system put together. The only thing that we tried, just the baits, no vaccine in them, just the baits. With respect to the production of the vaccine, we are in the final phases. It will be produced in white blood cells and in stable cell lines. But what we want is to go directly to the MA104 directly. Today, we can produce it in leukocytes, but we don't think that's the most appropriate thing. So that's why we're going to the cellular line to MA104, which works very well. We need this vaccine to be replicated, so it will probably be produced massively in BSL-3, and we will have the refrigerated and the frozen versions. So the main conclusions, and with this I will conclude, and of course we'll move to questions or comments. We already have a very good candidate for a DIVA vaccine. It's the Delta AB and others that are being evaluated, which I described, Delta AB I8. For domestic pigs, we are working with other mutants as well and making adjustments. This has been proven in wild boar and the AB I8 as well, but we are doing more assays and tests on domestic pigs. The doses and the administration pathways are important factors that make a big difference between the domestic pig and the wild boar. Remember that it is intramuscular for domestic pigs, and it's simpler, obviously, than orally, but we cannot vaccinate wild boars. It's impossible to vaccinate them. It's impossible to go out there in the field and vaccinate them. So we can do it orally because they're very attractive to the bait. It's really amazing how they go after the bait when we do these tests, like I said, with no vaccine, but to see how attractive these baits are for them. There is an ELISA test and the kits that have been already developed for the differential and determine where the vaccine is and where it isn't. And just a reminder, please, even with a vaccine that is commercialized, even when we have it, never forget that the best vaccines that we have is biosafety. That is the best vaccine that we can have, and we must focus on that. And if they have been, I mean, all of the areas that have been delimited. So the designs that we have for the vaccines are not really massive. They are almost therapeutic. When we see an area that could be isolated or that could be cut in advance, these are strategic. And we do not think that it is necessary to have general vaccination unless the country is really covered or plagued with the disease. These are my acknowledgements. Thank you very much, Ika, for inviting me and for spending this time together with all of you. Is always excellent. Those of you who know me, I love your countries. I love spending time with you. Every time I can, I travel over there, and I'm very happy to have accepted this invitation. Of course, thank you to all the colleagues of Vacdiva for the excellent work that is being carried out right now, and the ICA and the organizers for inviting me today, and to all of you for your attention, for your encouragement, the encouragement that you send me all the time via email, and the affection that you always show in your countries. So thank you all. And of course, I am at your disposal for any questions that you may have. Thank you very much. Thank you very much, Dr. Vizcaíno. Your presentation was really excellent. And thank you, actually, because you are always willing to collaborate with this side of the hemisphere and to share your experience and with everything that is happening in Europe and Africa. So thank you so much for telling us how things are going. I don't know, Dr. Vizcaíno, I'm not sure because maybe it's very late there, so maybe it's better to have the questions now, or should we wait for the end? What do you prefer? Whatever you prefer. You're the boss, Erica. You're the boss. If we wait until the end, maybe it's better with the three panelists. Yes, so we will leave you for a few minutes, and then we will pick up later. Excellent. Then we will continue with our next speaker. Our next speaker will be the experience of Brazil, and this will be in the hands of Alejandra de la Cerda Alvarez. But before, just a brief description of Dr. Alessandra. She is a veterinary doctor. She graduated from the Federal Rural University of Rio de Janeiro, and she works in the Ministry of Agriculture and Livestock of Brazil since 2007. She worked in the area of inspection until 2012. She joined the Animal Health Service of the state of Santa Catarina, and since 2019, she has worked in the National Program for Pig Health and in the Virtual Technical Unit of the Department of Animal Health. So after this brief presentation, Alessandra, you have the floor and you can share your screen.
Boa tarde a todos. Primeiramente, queria agradecer, muito obrigada, Érica e ao Ica, pelo convite.
Thank you, Erica, for the invitation. It's a pleasure and an honor to be here with you guys so as to share with you Brazil's experience in connection with ASF, African Swine Fever. So I will now screen share.
Já está aparecendo?
Can you see my screen right now? Not yet. Okay. Okay, we're good now.
Obrigada.
Thank you.
Então, desde 2021 que nós demos
Since 2021,
sistema de vigilância
we launched a new surveillance system.
clínica clássica e a partir de agosto de 2021
In the past, we used to have a system for classical swine fever, CSF, and in 2021, we launched an integrated plan for the surveillance of diseases in swine. So, okay, the new system involved reviewing the prior system, and it broadened the scope of surveillance for ASF and the respiratory reproductive system syndrome. So this is a system that is applied to the entire areas affected by classical swine fever, and its purpose is to do early detection of these diseases of CSF and ASF, as well as the other disease that I mentioned. So this plan was based on an FAO surveillance plan, and it considers the various realities for risk and production that exist in the CSF-free area, as well as the risk factors that are characterized by farm type. In this way, sampling is addressed to sectors with a greater likelihood of detecting these diseases. This enables greater sensibility or sensitivity of the system while using a smaller amount of samples. So right here, this entire area that we can see in brown, this is the non-CSF free area, whereas the green areas, you can see that there are different shades of green right here. These are CSAF free areas. So the area or the region was divided into areas 1, 2, and 3, depending on socio-geographic differences, and also in connection with the production or based on the production systems, the differences in production systems in these areas. So we checked for the prior likelihood of CSF presence in each area. So we made sure that the disease was absent in these areas. And also we based our criteria on the proximity to the non-CSF free areas. Characterizations were made based on types of farm, that is to say, how were the pigs raised. So this was a PhD paper that involved evaluating the surveillance system for CSF in the state of Santa Catarina and Rio Grande do Sul. Both states are located in the south of the country. So for CSF, we have 3 disease-free zones in Brazil, where we obtained the disease-free certificate by FAO. Well, this requires ongoing surveillance so as to show that CSF is absent in the disease-free zone. With regards to the PRRS, this was never detected in the country, and this requires ongoing surveillance for early detection, enabling fast containment in the case that the disease is detected. In the case of CSF, we did have the disease. It was introduced in 78 in Brazil, and it was finally eradicated in 84. From then on, the country has been considered CSF free. So this requires ongoing surveillance for early detection and fast response in the case, if there is a reintroduction of the disease. So, well, our surveillance plan is based on 5 components. Component 1 is based on serologic surveillance based on risk. Then we have inspections in the breeding houses or the breeding farms, research or investigations for suspicious cases, inspection in slaughterhouses. 5, serological surveillance in feral pigs. And our target population is as follows. We've divided the pigs into technified and not technified areas, that is, say, farms with or without technology. With regard to technology fitted farms, we have a production units for swine or for pigs, and we also have certified farms. These are farms that receive a certificate to be able to market the produce elsewhere. The pig keeping areas were not considered because they are considered to belong to a lower risk category and because the pigs do not spend too much time right there. For non-technology farms, we have local farms and survival subsistence farms. With regard to domestic farms, we also consider feral pigs. With regard to component one, serologic surveillance based on risk, well, this is the second cycle of our plan. Cycles start in July and they end up in June of the following year. The second cycle kicked off in July 2022 and it lasted until June 2023. We can see a map right here with the various farms that participated in the sampling. In pink, we can see the non-technological farms. In purple, those that involve the full cycle, and in green, the pig producing units. So this design was to have sensitivity worth of 95% and the likelihood for CSF absence equivalent to 99%. We did sampling for each state of Brazil. So there was a total number of sampled farms divided between the three categories. Non-certified farms were not included in the sampling because they are part of a regulation that provides for carrying out regular tests, that is every 6 months, to get a renewal of the certificate. So they follow differentiated sample in that case. So blood samples were collected in these farms, and based on the official vet service, the farms were visited to perform a general infection in the farm, as well as the clinical inspection of the pigs that was also performed. These were the pigs that were included in the sample. The pigs were sampled in order to detect clinical signs and symptoms. For the second cycle, we have almost 1,600 farms and almost 1,800 samples that were gathered. All of them came out negative for CSF. As part of this surveillance effort, we are just testing CSF, classical swine fever, in connection with component number 2 of the inspections that were performed in the nurseries or in the breeding farms. Sorry, this is part of active surveillance. There is sampling as well, and the official agency visits the farms in these three categories. On the map, what you can see in red, these are the full cycle farms. In yellow, the non-technology swine breeding farms, and finally in the purple, the pig production units. The official service performs a general inspection and clinical inspections across a number of individuals in the property so as to assess the animals and make sure that there are no alterations in the zootechnical indices, which might be indicative of hemorrhagic disease. So there is a protocol of activities that is followed. The official service fills in a form and risk factors are evaluated for the outbreak of the disease. For example, evaluations are made of structural, production, and health-related aspects of the farm, of the breeding center. In total, the inspections covered more than 1 million pigs. Now, in connection with component 3, which is the investigation of suspicious cases, that is part and parcel of passive surveillance, which is the main component of the system because it enables early detection of the disease. The service receives notifications for signs that are compatible with the disease, or, for example, whenever abnormal mortality is noticed. This is done by private laboratories and research institutions because they are generally looked for by private institutions so as to diagnose diseases in their properties, and they can come over to detect lesions or signals that might be characteristic or suggestive of the disease. In addition, and in order for this component to be effective, it's very important that there should be educational actions. That is to say, people should be educated on animal health, and the general population should be, there should be awareness raising efforts amongst the general population. So there need to be communication channels for general population, breeders, vets, that is every sector in the production chain should be involved when it comes to awareness raising. Well, we have the Regulation 50/2013 of the Ministry of Animal Health. This legislation establishes that if there is suspicion for CSF or ASF, it is necessary to act on any suspicion, especially for someone that works in domains such as diagnosis, teaching, veterinary sciences, etc. That is to say, whenever there is a suspicion, they're obligated to make a notification before the National Service if they suspect the disease. Our system is called CISBRAVET. This is a veterinarian emergency service that enables recording notifications by using this link. This means that anyone can access this link just by one click. They can make a notification or post a notification. So that is one of the ways in which they can do it. They can also call a phone number, and they can also show up at one of the offices. So whatever means is more accessible and easier to use for each citizen. In connection with passive surveillance, we've decided that we received 30 notifications that we considered to be valid, and out of those 30 notifications, 22 were ruled out as part of the initial investigation because it's not. Because we visited the farms, our national service visited the farms, and no signs or symptoms were detected. There were no especially high numbers of mortality. So 8 of the farms were classified as potential cases because the national or the official service did confirm signs and symptoms that were coincidental with the diseases, ASF, CSF, or PRRS, but the results came out negative for all 3 diseases at the end of the day. So with passive surveillance, all the samples are tested for all 3 diseases, ASF, CSF, and PRRS. In total, they analyzed a total of 31 tests. An ELISA test was done for ASF, for CSF. PCR tests were used for CSF as well. 94 samples were isolated, and these tests or these samples were isolated. All of them came back negative. With regard to component number 4, which is inspections in the slaughterhouses, during the inspections carried out by the official veterinary service in the slaughterhouses, before and after the slaughtering, it is possible to detect clinical signs and symptoms that may suggest, that may be suggestive of a number of diseases. And this may enable us to carry out actions so as to fight those diseases. In the case that the official service should detect lesions compatible with CSF, ASF, or PRRS, they will need to get in touch with the animal health service so as to perform a clinical, epidemiologic, and laboratory investigation in the farm where these cases were detected. Those inspection data can also help confirm the absence of diseases, if that should be the case. So during the second cycle, the total of pigs that were sent out to the slaughterhouse was of almost 60 million. 60 million pigs were sent to the slaughterhouse. Component number 5, which is serologic surveillance in feral pigs. This surveillance was done together with the official vet service, and this was done with the population management agents. These agents are individuals that are enrolled in our environmental agency, and they're also enrolled in the veterinary service of the National Veterinary Service. And these guys offer their collaboration with the official service whenever wild boar is hunted. And well, what they do is they do blood collections and they send it over to the official service. So this these population management activities are coordinated by the Environment and Natural Resources Brazilian Institute. IBAMA is the acronym, and the objective is to reduce the population of species that may harm the environment. We have surveillance protocols if there should be if there should be a detection of positive or inconclusive cases. These blood samples are tested by using an ELISA test. This is for CSF. And as I said before, population management agents are enrolled and trained so that the blood should be properly sampled and collected so that the samples may be analyzed. The total number of samples analyzed was 1,595, and well, you can see the number. There were 5 samples that came back positive or inconclusive using an ELISA test, and we used a screening test, and whenever the samples came back not negative, they were tested once again using virus neutralization. So the samples that were tested for viral neutralization came back negative as well. As soon as we have a result that is different from the negative result of ELISA, the official service is notified and a protocol is followed so as to carry out an investigation in the relevant farm so as to find out whether there are clinical signs of hemorrhagic diseases. It didn't happen in this case. Apart from this comprehensive surveillance plan, we have other measures for the prevention and containment of ASF. We can see some of these measures on the slide. Like, for example, there is this regulation that bans pigs from remaining in the vicinity of drinking troughs, as well as they also ban the picking up and the use of food remains or food remnants in these places to feed the animals. Also, the slough, the drinking troughs are inspected to detect the presence of animals. We also have import requirements for pigs and swine genetic material. We have a quarantine station prior to introduction, so the animals are quarantined right there before they are introduced. this is for imported pigs. That is to say, the pigs are quarantined in their country of origin, and when they arrive in Brazil, they are switched over to another quarantine station, and new tests are carried out. So as to detect the presence of ASF and CSF, as well as other diseases. We also have the VG Agro service. This is the agricultural and breeding surveillance service, which takes place in the ports and airports. There are detector dogs that are used, dogs that are specially trained to detect the disease. We also have a protocol for waste disposal. For example, in some cases, the entryways was through food remnants from a ship. So as a result of that, we launched this work that involved eliminating or disposing of that waste correctly, so that all risk from pigs might be eliminated. In addition, inspections were carried out on the boundaries or on the borders, and apart from that, the communication strategies were adopted. I will explain these a little later. We have a prevention plan for ASF, and we also have a contingency plan. This is thanks to strategies and procedures that have been implemented for containment and eradication of ASF outbreaks. And we also have had drill exercises for vet urgencies.
Aqui tem umas fotos, né, só para exemplificar para vocês, exercício simulado que foi realizado no estado de Santa Catarina em 2022.
Just so that you can see, this is a simulation exercise as was done in Santa Catarina with more than 180 professionals, both from the official veterinary service of Brazil and the 27 units of the Federation, and also a private initiative in other countries of Latin America.
Aqui para exemplificar uma das estratégias de comunicação utilizadas, então foi elaborado esse livro, Diálogos para a Prevenção da Peste.
Here, based on these communication strategies, we developed this book as an initiative of the Education Commission of Sao Paulo and the Department of Health in the state of Sao Paulo. It was developed by a multidisciplinary team made up of professors, researchers, and professionals, and basically for other backyard operations. And this is to teach how to prevent ASF. So this would be replicators of this scenario. We have this addressed to other audiences, other materials. In the Ministry of Agriculture, we have voice messages, infographics, posters, and other messages shared via WhatsApp. Another campaign that was developed called Swine Fever Not Here. Brazil is counting on you. African Swine Fever Not Here. This was a campaign organized with the private sector and as well as the National Union of Agricultural Auditors. And there were a series of cards developed with a great level of dissemination on social media and the private sector. Another campaign was valid, and the ASF campaign that was carried out to avoid it from arriving in Brazil. It included advertising on airports and ports, as well as dissemination on radio and TV channels. We also had Q&A, like frequently asked questions for travelers, for veterinarians, for the public sector, and the general public as well, and also for pig farmers and hunters as well. All of this material can be found here on this link that appears on the slide. With respect to the prevention plan for African swine fever, here are the specific objectives that were created, including improving pre-border and border actions to prevent the entering of the ASF virus, mitigate the risks of exposure of pigs to the infectious agent, strengthen early detection of suspicious cases, improve and expand the capacity to diagnose the disease in a rapid and precise manner, improve preparation and capacities of response in the face of animal health emergencies, and finally, creating conditions for the continuity of the business and minimizing commercial impacts if there is an introduction of the disease. This was created based on a methodology by defining operation matrices for each of the specific objectives. And all of this was developed along the process. And to exemplify the actions that were developed, these are in the phase of being adopted, and there was a study to determine the risk factors of ASF in Brazil, to analyze the occurrence of the disease, risk factors, etc., in transborder activities or transboundary activities. Of course, it included implementing an operational system for risk management and for irregular international transit, implement a software to manage data, including passenger information to identify profile of highest risk, survey the presence of infectious agents in confiscated products, permanently update the requirements of import for pig products and inputs, also reinforce procedures for quarantine for those pigs, and as I mentioned before, and finally define the criteria to suspend the import of products of animal origin. So this is for transboundary diseases, and for boundary actions or actions carried out at the border, we can include the following: implementing sanctions for the irregular transportation of animals, products, and subproducts, implement the national surveillance programs and agricultural defense in international borders, a program called Vigie Fronteira, structure the natural center for detection dogs. We want to include the use of these dogs, also establish a form, a declaration form for the luggage that is being carried, specific for the Ministry of Agriculture, intensify communication actions, increase the operative capacity of MAPA to increase supervision on international agriculture in general, implement sanctions for non-compliance with the solid waste management plan, which is mandatory for all companies that manage ports and airports. They need to submit a plan for the official service, and so what they do is audits on the execution of their plan, allow the adequate destruction of solid waste coming from aircrafts and also other forms of transportation, so the adequate way and the methods to appropriately destroy solid waste from aircraft and other vehicles. Prohibit the use of pig products when they are offered aboard the aircraft. So there is a proposal to forbid pig products on the aircraft. Analyze the data for imports and reinspection of imported raw material, to also do a proper treatment of the gut of the pig, regardless of its origin, and intensify the exchange of information with neighboring countries. These are the actions to mitigate the risks of exposure of pigs to the infectious agents, and this includes intensifying the supervision of subsistence pigs and raising awareness among farmers. Secondly, improving the biosafety conditions. As Dr. Vizcaíno mentioned, biosafety is paramount for the containment of ASF. We must also take measures to effectively prohibit pig farming in landfills, identify areas with presence of feral pigs, and regarding the previous point, to take measures to completely forbid the farming of pigs in landfills. Also identify areas, like I said, with presence of feral pigs. And the state services must ensure that there is a mapping of the different areas to see where these feral pigs are, and also to intensify actions of control for the population of feral pigs. Finally, educating these agents and bringing them closer to the official systems so that they can contribute to the integrated serological plan. And finally, improve the implementation of good agricultural practices in order to improve early detection and the protection of suspicious cases, probable cases, and confirmed cases. We have a technical file on ASF to have it very clear what is a suspicious case, what is a probable case, and what is a confirmed case, in improving the sensitivity or how sensitive the passive surveillance system is. Finally, improving the response of the official veterinary service to notifications when there is a suspicion. With respect to what we have found during the execution of this new surveillance system, and with respect to these measures for ASF, I would say that the greatest challenges are the following. The lack of human and financial resources. We do have some states in Brazil that do have these difficulties. The updating and appropriate characterization of the facilities that deal with pigs. Yes, this in order to take this sampling both for surveillance as well as for clinical surveillance. So all of the units in the federation must have a registration that is up to date with very good characterization. Another challenge that we face is to achieve the participation of the private sector, because in some regions where we see that pig farming is not very significant, it is more difficult to include the participation of the private sector for them to collaborate, especially to collaborate in the execution of the integrated plan. Finally, as the last challenge, which I believe is a worldwide challenge actually, is to define strategies to control feral pigs. As success stories for the 2 cycles of the plan, I can mention the results here that we have reached so far, which allow us to confirm the health status of the country with respect to areas that are free from CSF and free from ASF and free from PRRS. Also, the commitment made by veterinary services at a state level. Despite their difficulties, we were able to verify that the human resources, that means our colleagues which are in the state services, were deeply committed and made significant efforts for us to be able to fulfill the 2 cycles of the plan. Also, the cooperation of the producers was very important. There was no problem, really, with respect to not accepting the help of the official service, for example. The strategies that were carried out regarding communication showed that they are working. Actually, we saw that they are working and they are showing favorable results. And also the development of the prevention plan for ASF, which has the collaboration of different sectors within the agricultural sector and also outside the agriculture sector. So I think it was very successful to see that we were able to develop most of the activities and to really carry out this development. Finally, I would like to conclude with a few recommendations or advice. First of all, the fact that all of the surveillance system requires a legal framework. It is necessary to have a standard that provides legal support for the system. Also, maintaining a registration, proper registration of the establishments so that we can have an appropriate sampling design. The surveillance system is also must be designed in accordance to the human resources and available finance or funding, and make sure that it will not be fulfilled because of lack of financing. It is also important for the official veterinary service to have proper training, and this training must be ongoing. Another important aspect is that there has to be a commitment from different actors in the productive chain. For example, involve the different stakeholders is definitely a challenge and is key for us to achieve success in any surveillance program. So official services, the productive sector, veterinary services, and political representatives of the public and private sector are essential because they will be the ones who will define what can or cannot be done. So they must become involved and must be fully aware of the importance of surveillance. And finally, ensuring that there is a system to analyze the data that the surveillance system will be generating. This is what I wanted to present today. Thank you so much, and I hope that this was useful. I thank you very much for the invitation, and I would also like to thank you for having joined this session. I am at your disposal for any questions. Thank you. Thank you very much, Alessandra. Really excellent presentation, very complete, filled with details. Excellent. Thank you so much. And now we will give the floor to Dr. Soltero, and the questions will be left for the end. There are several questions already on the chat. I would like to remind everyone who is following us on social media just to send your questions via social media. Let's move on to the last presentation. Dr. Soltero has experience in Puerto Rico. And just to read his bio, Dr. Fred Soltero graduated from Kansas State University in 1977, where he received BS in biology and a doctorate in veterinary medicine. And following graduation, he started working as a veterinarian of the Puerto Rico Zoo in his hometown of Mayaguez in Puerto Rico. He started a small animal and exotic practice 2 years later and worked for 29 years before joining APHIS veterinary service. His specialty was avian medicine and orthopedics. He joined the VSPR as a field VMO and was interested in foreign animal diseases. And this gave him the opportunity to join the EMRS development team. and he later became part of an incident management team as EMRS specialist. He is currently the Deputy Incident Commander of the Green Team and has served in numerous deployments that include TB, avian influenza, exotic and virulent Newcastle disease, vesicular stomatitis, New World screwworm, etc. He has been involved in trainings for Incident Command System in Colombia, Ecuador, and Panama. He is currently working with IS on capacity training in the Southern Cone for HPAI, Southern Cone countries of Chile, Brazil, Argentina, Paraguay, Uruguay, and Bolivia, and is currently actively involved in the ASF protection zone in Puerto Rico. His regular work in veterinary sciences is as area veterinarian in charge for Puerto Rico since 2012. Very well. So I would like to give the floor to Dr. Soltero. The floor is yours. You can hear me, Erica?
Perdón, el tremendo. Bueno, muchas gracias, Erika, y a todo tu staff.
Thank you, Erica, and thank you to all the participants. I feel really honored. We are always invited to these types of webinars, and following Dr. Sanchez Vincaíno is no easy task. So I will try and cover what they have not covered, which is not much, but basically I would like to express why Puerto Rico is so flustered, because it's not that we are in a different country. We are 90 miles away from the current outbreak, 146 kilometers, that's it. And well, we need, of course, we should be concerned. Puerto Rico is a small island, but if you take a look at it, we have such a small distance, and there is an island belonging to Puerto Rico, the Muna Island, which is entirely inhabited by feral pigs, and this population is a wild population. There are different hunters that hunt right there, and of course it is banned for swine and swine products, but as you know, there are always people that break the rules, that are more than willing to break them. So from the Dominican Republic, especially in the East Coast, it is quite easy, while the weather is fair, in the sea, it's very easy to make a crossing from the Dominican Republic over to Puerto Rico. So there is a considerable risk, and that is why this area in the Caribbean has become the highest risk area. And that is why the USDA is investing so much to be able to protect us and to protect the US in general, not just Puerto Rico, from a potential entry of ASF from the Dominican Republic. Another factor that we are currently seeing in the news nowadays is the political and economic situation of Haiti. There is considerable conflict in the border with the Dominican Republic, and there are several people, even Americans, that are trying to leave the country. So if to the little surveillance that we might have, we add the lack of resources, well, that's a very serious situation. We are currently in the maximum level of alert, not just for Puerto Rico, but for the entire Caribbean. And this is something that we need to really take seriously, because we are getting to a point where if we do not take the necessary step, this is going to catch us off regard, especially in the Caribbean. So I'm going to move quite fast because I know that this has been discussed already. And of course, we have the Virgin Islands next to Puerto Rico. That's also a U.S. territory, just like Puerto Rico. And it's also a high-risk area because there are entryways through the Southern Antilles and there are entryways through vessels, for example, and the drug trafficking system. And so we are right in the middle of this entire risk area, high risk areas. In October, November 2021, with the ASF was confirmed in the US, straight away, it was determined that a protection zone needed to be established for Puerto Rico and the Virgin Islands so as to enhance all of the surveillance and protection and response systems that might be available. And you can see this in the numbers. So it really, well, we were caught by surprise, but the truth was that we needed to follow these instructions, that we needed to set up the system ASAP because between Puerto Rico, well, Puerto Rico and the Dominican Republic are two sister regions. We have a huge Dominican Republic. So we have a significant community of Dominicans living in Puerto Rico, and there's a lot of exchange, people traveling between Puerto Rico and the Dominican Republic. So these are factors that the prior two speakers mentioned as well. For example, Brazil is a country of many resources. Puerto Rico and the Dominican Republic need to work with the little we have so as to implement these types of protections. And of course, the protection zone establishes that we need to drastically increase biosafety. We need to enhance communication. We need to raise awareness amongst the general public. We need to increase, and I'm really surprised by the numbers that I'm seeing currently because we have an average sampling that we would have before the designation of protection zones, because before this was confirmed, we did between 1,500 and 2,000 samples per year for CSF, or classical swine fever, when ASF struck. And this is even more surprising for me because of the numbers that were shared by Brazil. Brazil did over 17,000 samples. I have just received 2021 information. So this, in order for you to understand that this is enhanced surveillance, Puerto Rico is 100 times smaller than Brazil, at the very least. So up until last month, we did 14,700 samples for ASF-CSF. So this is the enhanced surveillance that was afforded to us, and it's the only thing that we can do until new mechanisms may be put in place, such as the vaccine. I'm really impressed, actually, by the work by Dr. Sanchez Vicario, and I really hope that this success will continue because this is a tool that needs to be thought of thoroughly. So straight away, we needed to establish a number of mitigation measures. First of all, it's necessary to train the breeders, the vets, the general public, and nobody knew what ASF was because, well, it was viewed as a disease that came from Africa and that would never strike here. We had already had an outbreak in the 1970s in the Dominican Republic, and I know of vets that went out to help during this outbreak. But at that point in time, the Dominican Republic, together with the USDA, made the decision of total mass depopulation, and the US paid for a lot of those animals, and they tried to do the same in Haiti. But at this point in time, this option is not valid, or it's not an option right now for many reasons. So the mandate of enhancing surveillance considerably was something that we had to follow suit with, and surveillance had to be both passive and active. When Erica asked me about passive surveillance in Puerto Rico, well, we decided to go for a hybrid model. Our teams are ready to do passive surveillance and switch it over to active surveillance whenever necessary. And as with all passive surveillance in every country, it depends on having very good structure and a very good communication exchange. And well, the truth is that that is not so effective at this point in time in Puerto Rico. Right now, we do not have a proper information system, hence active surveillance, where we need to visit all the farms, no matter their status, no matter if they are commercial, no matter what their characteristics, they are to be visited, and there's a period of 2 to 4 months between visits. For example, I've seen animals that we have sampled with their blood over 15 times over the past few years. We do not have so many animals, but you know, it's enough with getting one contaminated product in between our borders, and that's it. So the sooner we make the detection, the easier it will be to preserve our industry. Of course, it's never easy, but we can have partial success if we act promptly. We also have surveillance having to do with sampling. Puerto Rico does not have a jurisdiction over the Virgin Islands. It's South Carolina that has a jurisdiction over that, but their surveillance program is enhanced as well. What we did establish, and I think that this hit a record in Puerto Rico, we used to have no laboratories that were able to do screening sampling, that is to say, at least a preliminary sample. And in just 3 months, we were able to create a satellite of the national US lab where, together with the US Department of Agriculture, who owns or which owns the lab, we were able to get going with the sampling efforts using the same equipment, the same technology, using employees from the USDA lab who came over to create this satellite lab. And it is in this way that Puerto Rico employees got trained. And right now, we are running the samples. Of course, we are not running at full capacity, but necessary tests are being performed at the same level and with the same quality, so that Dr. Marvin Holland, who is the new director and who spent some time last week here, was really happy about it, because we have the beginning of a potential lab that may be used even more in the Caribbean. So clearly enough, we are currently analyzing all the potential entryways of the disease. I'm talking specifically about ASF, of course, the entries into to Puerto Rico and the US. So the current situation that is being experienced in Haiti needs to be taken quite seriously, because we should, if you take a look at how difficult it is for all of the trapped people to leave the country, imagine the biosafety challenges involved. There are increased risks. There are people that are trying to make it over to the Dominican Republic in order to catch a plane and leave for the US. It's just amazing. For example, today we have had to increase the surveillance even more. So if we take a look at these targets or the goals for protection zones, importantly, if in Puerto Rico over the Virgin Islands there is an entry of a case and a case is confirmed, the US is going to try to isolate that. It will try to isolate Puerto Rico so that its industry and the rest of the United States are not affected, because basically the goal is to create a bubble thanks to these surveillance systems that have been set in place. And that is what we hope will work. If this is allowed in the new IOE, we're really concerned about up until what point, up until which point will we be able to continue with this high intensity surveillance efforts? Hopefully it will not be for long. As you know, you're well aware of the situation that in the Dominican Republic, and they have not been able to make the progress that they wanted to make. There are several cases that are detected on a weekly basis. The Vietnam vaccine test did not come out as successfully as they hoped for, so this tool is not available just now. And what we are concerned about in Puerto Rico is that we might see a point in time in which the country may give up, just give up, and the country might become endemic. So all of these AICA countries and ourselves are going to be faced with a serious problem, because hopefully we might be able to create a permanent barrier. The sea helps us, but there are ships coming into our borders. So, so we do have these series of activities. We have a program called the Illegal Boat Program, which is zone-based. It works 24/7, and the Coast Guard systems, the Border Patrol lets us know that there is a vessel that is on its way, that there's a boat that has arrived on our coastline. So what we do is we go straight there and we pick up all the products, no matter if they are meat products or they're plant-based products, because agricultural products are being brought over as well, and that can entail a serious problem for us. So this is accounted for, it is entered, the data are entered into a system, and all of these products are sent over to a privately owned incinerator, which is approved by the USDA, and this is done on a daily basis. Of course, there is not an illegal boat landing on a daily basis, but there are some specific days where several boats hit our coastlines. We have active surveillance for all swine premises. All of them are entered into a data system, which I discussed before. And at that point, they have been assigned, and the supervisors themselves do not need to even think about it. The owners have gotten used to it. But there comes a point in time in which the same people are coming back over and over again. That is why we are trying to find other tools, and we are trying to get started with oral fluids for several reasons. So first of all, it's a good option because it's non-invasive. Someone may be carrying out surveillance in multiple farms. This is not traumatic for pigs. It has not been approved just yet for the US laboratory system, but there are other countries such as Spain that have approved it already. So we are totally engaged with passive surveillance whenever there is a report of sick pigs, or whenever we arrive at a farm and we see a pig that may be involved. As we are hybrid with our system, passive surveillance turns into active surveillance straight away. An investigation is launched and samples are collected from tissue, from vessels, etc. So anything that may be necessary. Another, there is a test that is currently being used by Spain already. These are the new fast tests that are carried out on site. As soon as we can validate them, the better. For countries where labs are not, where a national lab is not available, that's a very good option because it opens up the door for very good diagnosis. So this is one of the possibilities that other island states may use, like, for example, even Central America. So we have slaughterhouse surveillance. It did not exist before. We have it now. All of these animals are bled in the slaughterhouse. We have fitted them with RFID identification, so we do know where those pigs come from. We have our sister agency, which is the wildlife agency. These guys sample all the pigs that they capture and that they slaughter. This program was in place before the arrival of ASF, and wild boar or wild or feral pigs have been eliminated by the thousands. So we have a ferry that travels twice a week from Santo Domingo to San Juan. Surveillance needs to be carried out there based on the loads, on the people involved, and so on. And well, no matter how much you may raise awareness amongst people, the sausages are always there. And this ship that had a very high factor needs to be helped out because the economy needs to continue to happen. Products need to be exchanged between Santo Domingo and San Juan de Puerto Rico. And our PPQ system is checking out on the mails or the postal service, the FedEx services, UPS, that is to say, all the packages that may arrive are taken care of. There is surveillance. And for example, there are also meat products that are sent over to the US. So there are too many doors that are currently open that we need to see to. So I was telling you all that we are doing. I'm sorry that this is in English because this presentation was created for my own national team. But well, what we've said is that we are currently performing intense surveillance and we're working with all our other possible agencies so that they may help us. Up until what point? Well, until we run out of resources and in the consideration of the fact that we need to continue to try to find new tools. I'm like a kid in a candy store right now because of this Diva vaccine information. It's really got me really excited. So I really would love for this process to move forward. So if we compare well, we've done quite a bit of sampling if we take a look at these numbers right here, and if we take a look at what Brazil has done, I'm really proud of what we have done ourselves. This is the average before we got started with surveillance. And the difference from one year to another. And well, you can see the figures right there for such a small island. That's quite a bit of samples. So we're really happy. But for how long can we continue to perform this type of surveillance? Well, whenever it's for so long as is needed.
Dorado is able to do PCR and ELISA testing in serum and whole blood. We have PCR to do ASF and CSF in a molecular transport media that's probably being used in Spain. This is one of our new toys that we're playing with, where we use it and it inactivates the virus as soon as the virus touches the media. So the sample is no longer infectious and all the material is preserved in order to do the PCR test. It does not need any kind of refrigeration and it lasts about 1 week until it reaches the lab. So this form of transportation for the Caribbean is really one of the new tools that we will be exploring. And we have been using this since December of last year. And everything has tested negative against all other tests. So we trust the MTM significantly. We also have to gather all the tissues, and when we are doing the research, we have to use the spleen, the tonsils for the virus isolation. And we're starting to use oral fluids in FAD investigations. We don't have permission to start active surveillance, which would be ideal, really, because there you reduce the number of employees and you have a lot more samples per animal, because I don't know, for 25 pigs, you can test more animals than with a traditional sampling method. So we're doing PCR and ELISA in all of those samples. I know that you want to go have lunch, so I'm going to go faster. This is in Puerto Rico. We have 12 areas of work. I know that Puerto Rico looks small, but the topography and the traffic is horrible. To reach one side of the island to the other, it is sort of half a day. So to work around Puerto Rico, you have to have options and vehicles that are prepared for all those mountains and hills. And I only have 3 VMOs for the entire country. And as you know, they're not always available. Yes, sometimes they're on vacation or doing something else. So there are no unlimited resources. And in Puerto Rico and the Department of Agriculture allow us to give food, but in order to share this waste, you have to have a license. As you know, the transmission of ASF and CSF through food is very, very dangerous. If it were for us, we would forbid it, but we can't do that because the Department of Agriculture and that amount of food do not have like a. But if there was an outbreak or if there was confirmation, this would have to be considered immediately. We have other farms, although they are not very big. Supposedly, they don't cook, but they do cook, and they're not in the system. So we need to look into them as well. We have like 20 farmers, which you call stratified, or those who have good biosafety procedures. Those are, of course, as protected as possible, and that's where we're processing the licenses. Erica, I think I will jump quickly through this. Yes, just because Dr. Vizcaíno has to leave, so that's why. But don't worry, finish your presentation. No, I want him to have time to answer questions. So these are the areas where illegal boat landings affect the Dominican Republic and Colombia. So you can see very clearly. And so all of the animals that are around those circles have to be controlled. Thanks to the data system, we can have the name of the farm and all of the information that we need. This is just the products that we are always seizing, which are weighed, identified, and it's as if we were doing a criminal investigation, yes? And this goes directly to the processing. We need to continue improving. We need to continue improving communication. This is improving every day, and we are doing more inspections. We have a space which are private travels, fishermen, boats, and those that go supposedly for pleasure between the Dominican Republic and Puerto Rico. Those are not being inspected. So I think that the possibility of having something come in is really not non-existent, yes? And we are a multi-agency effort. We work in collaboration with so many agencies that are giving a hand, but these are limited resources always. So I'm very grateful for the surveillance system, for all of the agencies that are helping us and who are helping us with the sampling as well. And this is in rural areas. So what the project is to implement oral fluids, because that increases surveillance significantly. And we must see whether the personnel resources can be followed, it can receive follow-up, and the possibility of the vaccine, of course. We are helping the Dominican Republic not only with training, but also we have helped them to We have done a project where there was a small cut on the ear for the bleeding, and which was much more quicker than from the neck. It's worked very well from the ear. So that's all. We're very concerned about the situation in Haiti. We are paying close attention to international flights. I think that there was one flight there was being analyzed, and we will be doing an exercise, and I will invite Ika and Erica to do it, to see how Puerto Rico will be responding this. And this exercise will be done in May. There is no, I mean, the state is responsible for this. So what I'm mostly concerned of, and with this I will conclude, is that we need to help the Caribbean countries to receive training and to really think seriously about implementing a program, both regional or local, where if I don't have a lab, I have to have a nearby country that will help me. No country can think that someone will come in 2 or 3 days, will save them from a possible outbreak. You have to have your own plans, and you have to do it quickly. And of course, many of the countries that are here today need to start looking for samples that are taken quickly from the field, and to have a high level of sensitivity so that they can use them and say, well, maybe I have something suspicious, and so that they have some kind of equipment to respond to that. But we have to work together to achieve that. And Ika has been sending this message for years, and the message is a good message, and it has to reach the ears of those who must respond. Sometimes there's no response, and it's frustrating. But, we are so close to Santo Domingo, so close to the Dominican Republic, that we are scared, and we don't want ASF to come to Puerto Rico. Sometimes we cannot help it, but that's all. Thank you very much. Thank you to Noelia Mogeno from the other agency. Sorry, Erica, for taking so long, and that's it. Thank you. Thank you very much. I do apologize for rushing you. Dr. Soltero, it was very interesting to hear your presentation. The problem is the time zones, yes, that we have with Spain. That's actually the problem for Dr. Vizcaíno. It's already nighttime. So we will start with some of the questions for Dr. Vizcaíno so that he can go rest. Thank you very much for being here and staying so long and waiting. And I know we've held you here, so we will quickly start with some of the questions so that you can leave soon. So Dr. Campuzano and myself will be asking the questions, and we will start now with the first one. For Dr. Vizcaíno. So, Dr. Vizcaíno, this is from Ruben Gutierrez Arizaca. He asked us, what is the basic reproductive number or reproduction rate, R0, for this disease? It's lower than for foot and mouth disease because the disease is transmitted quickly, but not as quickly as those that are transmitted by aerosol. So the RO is average. Perfect. Thank you. Rocio, I don't know if you want to take the next one. Yes, of course. Of course. Thank you, Erica. Next one is for Dr. Sanchez Vizcaíno. And what is the impact of ASF in the wild animal populations in ecosystems in general? What measures are being taken to control and surveil ASF in wildlife? This is from Dr. Cynthia Gonzalez. Thank you. Thank you very much, Rocio. The problem that we're having in Europe is mainly due to wildlife. And one of the things that we're doing the most, although it may not seem true, we're trying to educate the population that goes to national parks and goes to rest areas so that they do not leave food there. Because the main problem that we're seeing, strangely enough, is that in the areas where they're living, there are areas that are visited by travelers or tourists, and if it's close to the highways, even more so. So many foreigners just maybe stop there to eat something because there are rest areas with benches, etc., and they leave a series of waste. They leave waste behind, and it is affected by the high circulation of people there. So we are more concerned about educating the people and to protecting the wildlife there, even more than to really have an incidence on the animals. And something else that we're seeing very often is that, of course, in the wildlife, the thing we do in general is avoid the carcasses from remaining in the areas where those animals have died. And we do that with what is available to clean in each area. So two things: cleaning the carcasses very quickly, because only one carcass that died because of ASF, the number of animals that could become contaminated is very, very high. So that's the most dangerous hotspot. And finally, the food left behind by tourists, which is very concerning. I don't know if you've seen it on TV, but we had an outbreak in Rome, next to the Vatican, of a dead boar because they had eaten the rest of eaten things that were left in a trash bin. So unfortunately, disease is present in many, many countries, in part because of this. Thank you very much, Dr. Vizcaíno. Next question is also for you. What are your recommendations for veterinary services that are only now starting to prepare the epidemiological surveillance for ASF, considering the few resources that are available, both human resources and financial resources, so that actions can be more efficient and the lessons can be better learned? This is a very difficult question, Erica. I'm sorry, but it's combined so many things. We have to reach a large number of people. The more, the better. Because if you can understand what I said before, one of the greatest problems that we have is not with professionals, with farmers, but it is the general public. And it is difficult to send that message. We have organized many, many campaigns, and what we're most concerned about is that the people must understand the risk that there is and what are the factors, the risk factors. In many cases, no one thinks that bringing a sandwich from their country and they're eating it happily, not many people know that this can constitute a danger. They don't really understand it. They really do not consider that. So what we're most concerned about is dissemination in ports, in airports. That's where it works better. The worst thing are highways, the rest areas of highways. That is where it is most difficult to convey this information. And that is a problem. But we are mostly concerned about food being contaminated and arriving into the countries from a country that is infected. That's a great concern. Dr. Campuzano, go ahead. Rocio, I think your mic is off. Next one is for Dr. Vizcaíno as well. taking into account the difficulty of notifications by farmers, is it recommendable to do surveillance during the slaughter, and what considerations should be taken into account? It's a very interesting question, Rocio, because actually, as I said before, one of the greatest dangers that there is food, the food for human consumption of pig origin. So during the slaughter is a strategic point where certain risk factors can be eliminated. But an education campaign needs to take place for those who work in the slaughterhouses, yes, in the slaughter areas. This is very important because it could limit certain tissues that could eventually pose a problem. But in affected countries, this may be there, but the food can travel to countries that are not affected, and it can be transported by the people. So that's where there is a significant weakness. In slaughterhouses, the education is transmitted very well. It's very clear what they need to pay attention to, but the problem is travelers. That's the most complex problem.
I don't know whether I've made myself clear, Rocio. Rocio, you are speechless. Yeah, that's perfect. Thank you. Thank you. So the next question is also for you, Dr. Jose Manuel. I will not charge you extra for the questions, Rocio. Don't worry about that. So this is a question by Lina Mendoza in the risk or facing the risk of non-declaration of the disease. It would be good to know if in the EU there is a registry of exporting companies for processed foods towards the Americas so as to prevent the introduction of the disease right there. Yes, I think that there is good information provided by countries in Europe that export animal products over to other countries, not just pork, but other products. So I know that this is information that is easy to get. Thank you, Dr. Vizcaíno. Rocio, do we have anything to add? Yeah. For example, here in Mexico, the National Agri-Food Service, the Quality Service, has a registry of the plants that export over to Mexico, the factories. So if there was detection in a given European country, they are obligated to report that to the partner, to their commercial partners, so their commercial partnering countries. And in Mexico, you can consult this publicly available system. You can consult what plants are authorized to send over their products over to Mexico. And when there is an outbreak, the authorization of that plant is canceled immediately until the reverse happens. Yeah, it's the same thing here. All of the countries that are affected are not allowed to export, no matter what. And domestic consumption is tightly surveilled as well. It's very normal because it's a very significant dissemination mechanism, as a matter of fact. So it's good that care is taken to prevent that. The next question is also for you. That is why we would not let you leave, because there are so many questions for you. A question by Stalin Vasquez. He really loved your presentation, and he asks you, there is a probability of dissemination through raw materials such as feed, animal feed, wheat-based, soybean-based, maize-based, and what suggestions would you provide to prevent this type of transmission of the disease? Well, this is quite significant because there's been a significant amount of concern for that reason for a long time. As you can see, the grain is in the countryside. Animals walk by that place, by that area, and they start eating, etc. So we've seen that in these risk zones, the products are quarantined for a significant period of time so as to avoid or to prevent dissemination. So there's quite a bit of surveillance there, both in the silos as well as in the countries where the grain is bought in order to prevent infection, so that these are disease-free countries in as much as possible. And this is tightly controlled. And well, if the person that asked a question, yes, Stalin, it is a topic that has been dealt with for a long period of time already with significant surveillance efforts. Thank you, Dr. Vizcaíno. Yes, I think that this is a very interesting topic that needs to be addressed. Dr. Campuzano, please go ahead with the next one. Thank you. The next one is by Dr. Enrique Corona Barrera. He says that vaccination is strategic. However, once the disease is inside a country, the entire country is considered positive. Maybe the regulation might need to adapt to the circumstances. Yeah, well, in fact, that is what we try to do whenever health measures are required, like vaccination. For example, each case needs to be studied thoroughly. I agree with the person asking the question. Things vary quite a bit from one location to another because there are zones, there are regions that are affected, that are closed down entirely. So there can be no outlet of the disease at all. But it is true that each case needs to be studied thoroughly so as to ensure full safety, so that there is no dissemination. Usually for countries that are infected, the exports are completely closed down. So I think that this is a significant measure to be taken. I think that there are very good prevention measures. Thank you, doctor. And the last question, so that you can go ahead and rest. Yeah, well, dinner, actually. The dinner awaits me. Well, even more important than. Catherine Portilla is asking her following question. What factors do you recommend for risk analysis for the import of swine or pork subproducts or byproducts such as salami, sausages, etc., from a country where ASF is prevalent for domestic pigs or backyard pigs and feral pigs as well, where there is no zoning and where there is a free compartment of ASF and where there is the presence of wild boar? Well, this is no easy question. It's very difficult to control it, and the person asking the question has depicted a compelling scenario. It's difficult to leave that scenario, to make it out of that scenario. Hopefully there are not so many cases such as the one that he or she mentioned. This is a very challenging scenario, the one that Catherine described. Yeah, absolutely. Doctor, having said this, I would like to express my gratitude for all the time that you've shared with us. We will continue with the questions. Of course, you are invited to stay on, but there are a number of questions that we are going to send over to your email address because more questions are coming up. Yes, of course, I can answer the questions to you guys, or you can send me the email address of the person asking the question. But anyway, I will say my email to you. The email is jmvizcaino with a Z, Vizcaíno at UCM. It's the Universidad Complutense de Madrid .es. So that is the email. my email address. You can send me your questions. Thank you again, Erika. Thank you for inviting me to share time with this marvelous, amazing group of people. And I would like to say that I will always be glad to collaborate with you guys. So I send you my love. I send you my affection. I've always feel your affection whenever I travel in this region, and I always carry it in my heart. Thank you. Thank you a bunch, and see you soon. Yes, I would also like to say that everyone has left their greetings for Dr. Vizcaino. And just so you know, in the platform where we make these webinars available, we are going to leave you with Dr. Vizcaino's email address if you want to write to him directly with a question. Rocio, we have a question for Dr. Lacerda. Please go ahead. Just one sec. Doctor, well, this was already answered by you on the chat. The question is about the diagnostic algorithm for ASF or CSF and epidemiologic surveillance. Stalin Vasquez asks, how does sampling carried out in backyard animals, and how do you classify the breeders or the animal farmers in Brazil? Okay, thank you for your question. We carry out, we do differentiated classification by differentiated farms that have technology versus those that do not. Our national services have their registration systems, our registry systems, and when a swine property or premises are identified, they are classified. That is to say, if it's a technology-based farm, we'll have a full cycle right there, a pig production unit, a breeding center, etc. But if it's a non-technology farm, it might be like subsistence farming that is being carried out, or commercial farmer. When we do the sampling, we do it based on the registry of the federation. I don't know whether this was the question addressed to me. I don't know whether I understood it correctly. Yes, thank you. Thank you, Dr. Alessandra. The next question is also for you. It's from Rafael Nicolino, and he says, for these samples from ports, airports, and frontiers or borders, is there a protocol for carrying out tests on these products so as to detect a pathogen, such as the PSA pathogen? That is part of our agricultural surveillance service. Inspections are carried out for passengers and for their baggage. And for we have also import requirements. I don't know whether you refer to imported products. Is it imported products that you're referring to, or is it the products that the travelers bring with them? But well, for the imported products, we have import requirements whereby a certificate is issued with the official vet unit of the country. A certificate is issued that vouches that the importer is observing a number of requirements. And with regard to traveler products, they are inspected, samples are sometimes made, and for some products, re-inspections are carried out in the places, and this is under, the inspections are done by the federal sector, like the agricultural ministry. So there is a re-inspection that is carried out for pork products. Thank you, Doctor. Rocio, please go ahead. The next question is, are you interested to know, the person would like to know whether drills have been carried out with your neighboring countries or your main commercial partners? What results would you be able to share?
Eu apresentei ali coloquei ali slide na apresentação de exercício simulado que foi
I
feito em
presented
Catarina em
as
2022.
an exercise, a drill that was carried out in Santa Catarina in 2022 with the participation of neighboring countries. Another drill was carried out over the past 3 years in Mato Grosso.
A gente sempre procura convidar os países vizinhos.
We always attempt to invite our neighboring countries, and we hope that they will participate.
Esses exercícios sempre são muito proveitosos, né? Uma experiência
Those
que a gente vê que
drills are always quite successful. We've had a very good experience on that score because these are on-site drills, on-site exercises, and we carry them out as if the disease was taking place. We communicate with the CONAPRIM, which is the animal health agency. So we try to get all the stages or all the emergency phases involved. We get all the parties involved, all the teams involved. We also get the municipalities involved, civil protection. We also let the stakeholders know that the drills are going to be carried out beforehand, and the results have been quite successful. Thank you, Dr. Alessandra. The next question is for Dr. Soltero, and this would mark the end of our Q&A panel. Dr. Soltero, Dr. Orellana from Guatemala asks us the following. Have studies been carried out on the role of peccaries in the epidemiologic chain of ASF? Yes, my understanding is that this has been done. We do not have that species right here in Puerto Rico. I understand that the peccary behaves quite differently from other species, from other pigs. So I don't know whether it's really worthwhile to include it in this type of surveillance, but it is better to do the least bad thing. But I do know that the peccary behaves quite differently from the white boar. Thank you, Dr. Soltero. And well, this marks the end of our Q&A panel. For the benefit of time, we know that we have exceeded the time allotted to us by quite a bit. Thank you so much to all the speakers for their excellent presentations. Thank you for sharing this experience across countries. We are being joined here not just by the official sector, we also have members of the academia. We have members of private industry. We have Fit Latina, we have the official sector from Peru, from Mexico, from Colombia, the San Marcos University from the city of Lima. We have SENASA from Argentina, SENASA Bolivia. We also have the official services from Paraguay, Cuba. So these are some of the countries that have joined us today through the entire webinar. So I would just like to express our gratitude for all of the information that was shared, and we have received profuse words of congratulations for all of you. So, well, I wish you the very best. We will meet again in the coming webinar, which will be held in April and which will be on diagnosis. And I would like to thank my colleagues, Dr. Campuzano, our colleagues at AICA, and all the agencies that are part of this interagency group that makes these webinars possible. Please do not forget to visit our platform. You will find recordings of our webinars right there. We will try to share the presentations right there as well as today's webinars video. So thank you, Dr. Cassandra. Thank you, Dr. Fred. I hope you will have a wonderful rest of the day and see you next time.
Muchísimas gracias.
Thank you.
Hasta luego.
Hasta luego.